Okay, so let's talk about how and why the cell membrane in its construction is selectively permeable and allows certain things to go into and out of it. So on this plain white piece of paper, I'm going to draw a lipid bilayer right here. I'm going to leave some space on this side because I'm going to write some other things that pertain to this. All right, so let's just do a lipid bilayer like this. All right, so this is your phospholipid. Phospholipid bilayer. And I'm going to write quite a few things here, so I'm going to make it lengthy here. All right, we might have to add on if I didn't make it long enough. Okay, so really quickly, so pretend like this page is a cell. So the cell, this lipid bilayer would go all the way around in a circle if it was an animal cell. So lipid bilayer is separating the inside of the cell from the outside of the cell. So let's say this is the inside of the cell. Here's the cytoplasm. And the cytoplasm has a high concentration of water. These brackets mean concentration. The up arrow is just my shorthand for writing high. And then if this is the outside of the cell, the fluid on the outside of the cell is called the extracellular fluid. And this also has a high concentration of water. All right, so why the lipid bilayer is formed the way it is because of the water concentrations inside and outside the cell, and also because of the chemical nature of the two parts of the phospholipid. So this part right here, what is called the tails here, these are hydrophobic or philic? Hopefully in your head you said that these are hydrophobic. They're going away from the water. And these guys are hydrophilic. All right, where they're facing towards the water. Another way to say this is we could say they're also polar. We could say the hydrophobic is also nonpolar. Okay. So this is what we know. And then we also know, let's talk about why that is. So let's just review about the construction of a phospholipid up here. All right, so the phospholipid has, like we just drew it like this. So if I label this a phospholipid, nice shorthand like this, right? All right, but really, what is this head region? All right, the head region is a glycerol molecule, which is a 3-carbon molecule, glycerol, and then it has a phosphate group, and it has the tails here, which are the 2 fatty acids. So I'm going to put here FA, FA. So when we draw something like this right here is hydrophilic, this is hydrophobic. All right, this is the head region, this is the tail region, right? All right, so to relate this to this is, I'm going to use the same color, all right, hydrophobic. And this hydrophilic. Okay. So that's the lipid bilayer. So let's talk about how this chemical nature of the phospholipids determine what can get into the cell or out of the cell. All right. So let's start with, I'm going to use the color green. All right. So what kinds of things can get into the cell or through the lipid bilayer? So things diffuse going from a high to a low concentration. So if something is going to go into the cell, it can go right through the lipid bilayer, all right, like this, all right, if there's a higher concentration here than here. So what kinds of things are like that? And this is hydrophobic molecules. All right, so this, and the reason why is because most of the thickness of the cell membrane are the tails, which are hydrophobic. So hydrophobic molecules can move through this fairly easily. All right, so let's put in examples here. We could write oxygen, O2, can get N2. The reason why they're hydrophobic is they're nonpolar, because two atoms bonded together like O2 are going to be nonpolar covalent bonds because they're going to share the electrons equally. That means that one is not going to be pulling the electrons closer to itself, and so there's no charge on those molecules. All right, so hydrophobic molecules can get through here. There's a second kind of molecule that can get through here, so I'm going to draw my arrow here, another arrow. I'm going to do it in the same color because these are the molecules that can actually diffuse through the phospholipid bilayer. So we're going to write here small, comma, uncharged polar molecules. So examples of this would be carbon dioxide. So they're small, so size, they're polar, all right, so carbon dioxide, urea, that is a waste product that's found in urine, all right, would be an example of that as well. So you just need to know kind of the category. And so these things, if they're small enough, the key here, all right, I'm going to put in boxes small and polar. So small polar molecules can get through. Now, I said the thickness of the cell membrane is mostly hydrophobic, and that's why hydrophobic molecules can get through here. But if they're small enough, they found that if they're small enough, they can squeeze through there being polar, all right? So those are the two types of things that can get through the lipid bilayer. So these are not the only types of molecules. There are other kinds of molecules, but these guys can't get through. So I'm going to switch colors, and I'm going to switch to a nice purple color. So these guys, I'm going to illustrate these not being able to get in by doing one of these numbers. It's going to go hit the lipid bilayer and ricochet off. So showing you that it might want to go in terms of there being a high concentration on this side and want to move to the other side, but it can't get through that lipid bilayer. So what kinds of things would this be? So this is large, uncharged polar molecules. So large, polar. That's the difference between these two. Look at the side, they're both polar, but the large molecules can't get through. All right, so the size makes a difference. So examples of these are glucose, so sugar, sucrose. So this is a monosaccharide. Sucrose is a disaccharide. All right, they cannot get through the lipid bilayer. And there's one other category that cannot get through the lipid bilayer. So we're going to draw that here. We're going to do another ricocheting. And in this, it is ions. What is ions? Ions are atoms with a charge. So example, H plus can't get through. Sodium ions, potassium ions, all right, chloride ions. So you get the picture. Anything with a charge. And so these two can't get through this one because of the large nature of it, even though in its polar nature. These guys can because of its small size. And ions are just way too charged to get through that. The thickness of the fatty acids here, all right, so the tails there. All right, and one other thing, and it does look like I'm going to have to add on to this that I want to talk about, is that unique in itself is, I'll do one more. All right, so it can't get through, so I'm going to do here the ricocheting off here, and that is water. Water can't get through the lipid bilayer. And so this is unique, and it took them a long time. When I first started teaching, they thought water was in this category here because it's small and it's polar. It's only 3 atoms big and it's polar, and they thought it could get through the lipid bilayer, but lo and behold, they found that it can't. All right, and so how in the world do these things get in or out of the cell then if they can't get through the lipid bilayer? Because these things are needed. You need glucose in your cells. You need some ions for various chemical reactions. Sodium and potassium are super important in making your neurons of your nervous system work properly. All cells need water, but they can't get through the lipid bilayer. So how do they get through there? All right, so let's talk about that. So I'm going to put a little thing here for each one of these that can't get through. All right, the lipid bilayer. So let's write this. And you might want to think to yourself, how do they get through? And think about if you have an answer to that before I go over it. So these molecules cannot, and I'm going to underline that, cannot diffuse through the lipid bilayer. So that's what we just talked about. All right, so how do they get in? These are, and I'm going to use this word, facilitated into the cell by, here's our answer, proteins. Proteins, there are what we call channel proteins. They literally have a channel, or carrier proteins. They act to carry molecules across the membrane. So they'll use the word facilitated. If you facilitate something, you help something. And I wanted to use that specifically because of what this is called. This is called facilitated diffusion. I'm going to underline that, some super important word there. So these guys, lo and behold, can get through the lipid bilayer, just not through the cell membrane, just not through the lipid bilayer. There are proteins, and so there are proteins embedded within that lipid bilayer. Okay, and so this tells us what can and can't get through the cell and how it does that, if it can get through the lipid bilayer or protein.